Autoradiography is a powerful laboratory technique used to detect and visualize radioactive substances within biological samples. It plays a vital role in molecular biology, pharmacology, neuroscience, and medical research by allowing scientists to see where specific molecules are located inside cells, tissues, or even entire organisms. By transforming invisible radiation into a visible image, autoradiography provides a direct window into biological processes that cannot be observed using conventional microscopy. The principle of autoradiography is based on radioactive decay. A molecule of interest, such as a drug, DNA fragment, amino acid, or hormone, is labeled with a radioactive isotope. Commonly used isotopes include tritium, carbon-14, phosphorus-32, sulfur-35, and iodine-125. As these isotopes decay, they emit radiation. When a radio-labeled sample is placed in close contact with a photographic film or a radiation-sensitive detector, the emitted radiation exposes the film. After chemical development, dark regions appear on the film indicating the presence and distribution of the radioactive substance. Autoradiography can be classified into several types depending on the scale of observation. Whole body autoradiography is mainly used in pharmacology and toxicology. In this method, a radio-labeled compound is administered to a laboratory animal, usually a rodent. The animal is rapidly frozen to preserve tissue structure and then cut into thin sections. These sections are exposed to photographic film, allowing researchers to observe how a drug or chemical distributes throughout the entire body. Tissue autoradiography focuses on specific organs or tissue sections. Thin slices of tissues such as the liver, kidneys, or brain are prepared and exposed to film. This method provides more detailed information about where a radioactive compound accumulates within a particular organ. Micro-autoradiography offers the highest resolution. In this technique, a nuclear emulsion is applied directly onto the tissue section, enabling visualization of radioactive molecules at the cellular or even subcellular level. The procedure of autoradiography involves several carefully controlled steps. First, the biological system is exposed to a radio-labeled compound. This may involve living organisms, cultured cells, or isolated tissues. After sufficient time is allowed for incorporation or binding of the labeled molecule, the sample is fixed to preserve its structure. The fixed sample is then embedded in a suitable medium and cut into very thin sections using specialized instruments. These tissue sections are placed in close contact with photographic film or coated with a radiation-sensitive emulsion. The exposure time depends on the type of radioisotope used and its energy. High-energy isotopes may require only a few hours of exposure, while low-energy isotopes such as tritium may require days or even weeks. After exposure, the film is developed using photographic chemicals, producing an image known as an autoradiograph. Autoradiography has wide-ranging applications in biological sciences. In molecular biology, it is commonly used to detect DNA, RNA, and proteins after separation techniques such as gel electrophoresis. Classical methods like southern blotting and northern blotting rely on autoradiography to visualize radio-labeled probes that bind to specific nucleic acid sequences. Before the widespread use of fluorescent detection, autoradiography was considered the gold standard for molecular detection. In pharmacology and drug development, autoradiography plays a critical role in studying drug absorption, distribution, metabolism, and excretion, often referred to as ADME studies. By tracking radio-labeled drugs in the body, researchers can identify target tissues, accumulation sites, and potential toxic effects. In cell biology, autoradiography is used to study cell division and protein synthesis, while in neuroscience it is used to map neurotransmitter receptors in the brain. Despite its many advantages, autoradiography has limitations. Handling radioactive materials requires strict safety precautions, specialized facilities, and trained personnel. The technique can also be time-consuming, and radioactive waste must be carefully managed. However, modern developments such as digital autoradiography have improved image analysis and reduced exposure times. In conclusion, autoradiography is a fundamental scientific technique that allows researchers to visualize molecular events inside biological systems. By revealing the precise location of radioactive molecules, autoradiography has greatly advanced our understanding of biology, medicine, and drug development, and it continues to be an invaluable tool in modern research.